Okay, as for adenovirus, it is a DNA virus with broad cell tropism that can transiently transduce nearly any mammalian cell types. I mean, it's quite broad and can induce any cell types. Viral replication occurs without integration into the host genome, leading to only transient expression of the transgenes, which means that its expression is only for temporary use. But sometimes, for example, certain diseases, we don't want the virus to stay there. We want it to, the gene to be produced temporarily. For example, like cancer and angiogenesis gene therapy. The packaging capacity of adenovirus is more than 10 kilobase, which is larger than retroviral. The main advantage includes its efficiency for the transduction is high, as is the level of gene expressions, although this is only temporary and can be diminished rapidly within just a few weeks. But the disadvantage is that the adenoviral vector is really difficult to achieve cell-specific targeting since the virus has no envelope to attach to cell-specific ligands compared to the cell specificity achieved with retrovirus, which means that its engagement with the cell is too broad. It's not specific to certain cells. So it's not good, which means that it can also induce some safety concern, which means that what we want to deliver to specific cells may not be achieved with adenovirus. So this is just a representative diagrams on the use of adenovirus as gene delivery vector, which is quite common on how the first vector binds to the cell membranes and is packaged in vesicles, breakdowns, and then will inject the new gene into the nucleus to make a new protein. And then we have adeno-associated virus, which combine some of the advantages of both the retro and adenoviral vectors. It is a single-stranded DNA parvovirus that is able to integrate. Able to integrate means it can have a long-term gene expression into the host genome during replications and able to produce a stable transductions. In fact, a wide range of cell types, including both dividing and non-dividing. And good thing is that AAV are not associated with any non-human disease, which means that it is quite safe and show high efficiency transduction. But one of the obvious disadvantage is that it can only carry a fairly small therapeutic gene insert. Which is just around 4.7 kilobase, which is half of the size of the DNA that other virus can carry. And then we have lentivirus, which is belong to the retrovirus family, but can infect both dividing and non-dividing cells. And basically, lentivirus are able to integrate into the host genomes, hence being advantageous for long-term expression of the therapeutic transgenes, means it will not being. And HIV is a very effective antiviral vector because it has evolved to infect and express its genes in the human T cell, low immune response, which means that it will not be recognized by the immune response. So it has ability to avoid the immune response. So which means that the body will not produce any potent antibody response against the virus. So basically, it's really important for you to at least understand the differences between the virus. And for example, if we give you for the exam questions, like the disease and the requirement of the disease for the gene therapy, for example, it need to be long term or temporary, you need to know which virus to choose as the most appropriate vector. Okay, for example, if you want to have it to infect both dividing and non-dividing cell, so you should be able to tell which of the virus would be the most appropriate to use. All right. So this is a comparison on the specific and the differences between the virus that I've mentioned earlier. So have a read and see on the application, especially on what sort of disease that this particular virus should be appropriately to be used for. All right. Okay.